Hello guys, this is David Villar, and on this final video we will discuss toxicants that affect the peripheral nervous system. On this slide we have what substances this video will cover that include atropine and scopolamine, solanine plants, slaframine, organophosphorus and carbamate insecticides, nicotine plants and curare. Now to begin with, a quick reminder of the peripheral nervous system. All the nerves that originate in the brain and the spinal cord constitute the peripheral nervous system, which is divided into the somatic branch and the autonomous one, which is in turn divided into the sympathetic, parasympathetic, and enteric divisions. The somatic is integrated by only one neuron, whereas the autonomous divisions are constituted by two neurons. We see the same thing on this other slide. And what is important is to know about the type of receptors and neurotransmitters in each division. We can see that the somatic has just one neuron that originates in the CNS, and the neurotransmitter in the motor end plate at the skeletal muscle is acetylcholine that binds and activates nicotinic receptors. Both the sympathetic and the parasympathetic divisions have two neurons, with the first one relaying information to the second one in the ganglia. And the synapses in those ganglia use acetylcholine and nicotinic receptors in both divisions. However, the neurotransmitter on that second neuron will be different. Norepinephrine is released in the sympathetic division on effector cells, and it is acetylcholine in the parasympathetic division that will activate muscarinic receptors in different effector cells. So if we look at this other graph, we see where acetylcholine acts as a neurotransmitter. All the terminals of the parasympathetic release acetylcholine at the junction with effector cells. It is also the neurotransmitter in the adrenal glands that will release norepinephrine and epinephrine to the bloodstream. It is also present in the CNS, where it binds to either muscarinic or nicotinic receptors. It is also in the neuromuscular junction of the skeletal muscles, and is also in the preganglionic nerves of the sympathetic nervous system, and those going to sweat glands at the pre- and postganglionic neurons. And on this final review slide, it shows the actions of both the parasympathetic and sympathetic, and at each one of all the effector cells in different organs of the body. As a general rule, they cause opposite effects. In other words, if one slows down the heart, the other one is going to speed up the rate. If you notice, the ganglia where the first neuron relays the information to the second one is close to the spinal cord for the sympathetic division, and it is actually within or very close to the target organ for the parasympathetic division. Now we're going to talk about toxicants that are either agonist or antagonist of the cholinergic neurons, which are those that use acetylcholine. The terminology to refer to substances that block cholinergic terminals could be either cholinergic blockers, antimuscarinic, antiparasympathetic, cholinolytics, parasympathicolytics, antispasmodics, spasmolytics. And if you would like to revise where those cholinergic neurons are in the body, they are also repeated here at the bottom of the slide. So on this slide, we see the molecular structure of atropine and scopolamine. Both are tertiary amines that are not charged and can therefore penetrate well into tissues, including the brain. They work as competitive antagonists of muscarinic receptors, and that will prevent the acetylcholine from binding at those receptors. It will not do anything in the sympathomimetic preganglionic neurons or the neuromuscular end plates, because the receptors at those locations are nicotinic. Now, if you use butylscopolamine or buscapine, it is a charged molecule that will not enter into the CNS, and the effects will only be peripheral, like decreasing GI motility and spasms, And in fact, this is used as a drug for abdominal cramps, menstrual, and bladder spasms. Both atropine and scopolamine are natural tropane alkaloids. They are extracted from the belladonna plant and the henbane plant, and they are also present in other toxic plants like Jimson weed. Atropa belladonna is also called a deadly nightshade, and in fact, just the ingestion of 3 berries could be fatal to a small dog or a kid, for that matter. Here is a picture of Jimson weed that is extremely easy to identify for its big and simple leaves. They are irregularly toothed, and the flowers are also large and tubular. And the fruit is a spiny capsule that can be split into 4 parts, and as you can see, each one has a small brown to black seeds. Now, if we know where the muscarinic receptors are, we can deduce the type of clinical signs expected with an overdose of atropine. Because it penetrates into the CNS, it will have both peripheral and central signs. It's going to block the actions of the vagus nerve, so we can expect a rapid heart rate, urinary retention, constipation with colic in horses, dry mouth, mydriasis, loss of accommodation of the eye, so you cannot really see well up and close, but the distant vision remains sharp. Hyperthermia and some CNS signs may occur, like disorientation, excitement, tremors, and hallucinations in humans. Now, the treatment for a recent oral exposure includes activated charcoal and a saline cathartic. In a horse, you can give 1 gallon of mineral oil to dislodge the impaction, and Banamine IV for the abdominal pain or colic. For a severely poisoned animal, giving physostigmine IV to the point at which delirium and coma is abolished. You can give this slowly over 5 minutes, and the animal, if the animal responds, a diagnosis is confirmed. Now, if the tachyarrhythmias are unresponsive to physostigmine, they may respond to propranolol. Here we have the structure of physostigmine. As you can see, it's a natural alkaloid that competitively inhibits cholinesterase, and that prevents the hydrolysis of acetylcholine. So this is going to result in an elevation of concentrations of acetylcholine, which compete with the atropine for the cholinergic receptors. It is not a charged molecule like neostigmine, and that means it's going to penetrate into the brain. Now we're going to move into plants containing solanine. This is a glycoalkaloid that is bitter and very unpalatable. It is an esteroidal alkaloid, and it's comprised of an aglycone that is called solanidine that remains after the sugar moiety has been removed. And the glycoside, which is solanine, tends to be more toxic than the aglycone or the solanidine. The main clinical signs attributed are GI tract irritation and CNS depression. And the difference between atropine and solanine is not straightforward. They may look very similar in most clinical signs, but there are some clues that may tip you off to lean more towards one or the other. The main difference is highlighted in black. Atropine causes constipation, which is the opposite of diarrhea, which is often hemorrhagic. It also stimulates the CNS, which is the opposite of depression, and it also causes a dry mouth, which is the opposite of hypersalivation. All other signs are pretty much the same for both substances: dilated pupils, ataxia, weakness, thrashing movements, and hyperthermia. The problem is that many of these plants that we have here on the slide, which are all in the Solanaceae family, they contain both types of alkaloids. the solanaceous and the atropine-like compounds, and that kind of complicates the diagnosis and also the therapeutic considerations. You can find reports of intoxication in the literature by all these plants. If you want to remind something, the alkaloids make the plants quite unpalatable. So poisoning usually happens under conditions of overgrazing or when there is no better green forage available. Now, however, the toxicity is not lost when the plants dry, and that may render them a little bit more palatable because the sugar moiety is released. In other words, the wilted leaves are more likely to be eaten, and some of these plants are extremely toxic and may be deadly if ingested. Potatoes are in this family of plants, and particularly spoiled potatoes and the peelings have also caused severe poisoning, particularly when you see green parts on the potatoes. There is no specific antidote for solanine intoxications. If there are life-threatening poisoning based on clinical signs, you can consider physostigmine, as we saw for atropine ingestions. Now the next one is slaframine or slobber factor. This is an indolisidine alkaloid or mycotoxin. Actually, it's an alkaloid and a mycotoxin. It's often produced in red clover, which is infested with the fungus Rhizoctonia leguminicola. Slaframine gets converted in the body to a quaternary amine that is physiologically very similar to acetylcholine and works in exocrine glands, but there is actually no effect on ganglionic transmission, the heart, the blood pressure, or the neuromuscular transmission. So the first clinical signs, and the classical one, is excessive salivation. This may be followed by lacrimation, refuse to eat more of the contaminated forage, and sometimes in more severe cases you may see diarrhea and frequent urination. Now the most serious signs reported are abortion in both cattle and horses, and the diagnosis is primarily based on the clinical signs. SLUD is an acronym that is used to refer to salivation, lacrimation, urination, and defecation. As I say, in the majority of cases, all you see is salivation, which may be so severe in horses that when you tie them to a post for a few minutes, they make a puddle of saliva accumulating in the soil beneath their heads. Obviously, if you identify red clover in the hay and you see black patches on the leaves, it's usually enough to reach a diagnosis. There is actually a gas chromatography method that can also identify the slaframine in the hay. And with regards to the treatment, as soon as you remove animals from the source, they will recover. You may wonder if we could use atropine to stop the salivation, but in this case it's not very effective because slaframine has a greater affinity for the receptors. The next one is on cholinesterase inhibitors that are mostly used as insecticides. Most of the signs they produce are those of parasympathetic overdrive. And if we look at what the parasympathetic division does on every organ, you can pretty much deduce what type of signs an intoxicated animal will exhibit. Some of the worst warfare agents are organophosphorus insecticides like Soman, Sarin, Tabun. So you can imagine how toxic these are, and obviously these compounds are not used as insecticides. They are all synthetic compounds except for physostigmine, which is a plant alkaloid that we mentioned earlier, and that is used as the antidote for intoxications of atropine and scopolamine. So the toxicity actually varies a lot from one compound to the other, and it goes from very low to extremely toxic. Now, what are common sources of toxic exposures? The first one that accounts for about 80% of all the exposures in food animals is misidentification of granular pesticides as vitamins or mineral supplements. That is why you want to keep feed ingredients separate from pesticides when you store them. Another more anecdotal one has been residues in vehicles to transport pesticides prior to using them for transporting feed. And the mechanism of action, on the left figure we have a motor nerve and skeletal muscle fiber. When there is a potential action that reaches that nerve terminal, acetylcholine is released and binds to the nicotinic receptors in the muscle fiber. The acetylcholinesterase quickly hydrolyzes the acetylcholine at the location, and therefore the stimulus finishes. Actually, on the right figure there is an OP insecticide that is irreversibly binding to the cholinesterase, and this prevents the enzyme from breaking down the acetylcholine. And there is one big difference between OPs and carbamates. Generally speaking, OPs bind irreversibly, but carbamates do it reversibly. Now this is also explained on this other slide. OPs result in irreversible binding of acetylcholinesterase. Carbamates result in reversible binding, but it often lasts long enough to allow clinical signs, possibly death. The signs result from acetylcholine building up, and there is a continuous stimulation of the nervous, the glandular, the muscle receptors, whether they are muscarinic or nicotinic. So what are the clinical signs in poisoned animals? They can be assigned to either muscarinic, nicotinic, and CNS effect. The muscarinic ones include SLUD, that stands out for salivation, lacrimation, urination, and diarrhea. The smooth muscle fibers that circle the small bronchi will stimulate, and that causes bronchoconstriction. And on top of that, the airway glands release secretions which block those airways. At the heart, bradycardia will be more common than tachycardia, but because there is release of epinephrine by the adrenal glands, we may see tachycardia. And a common theme to most cases is meiosis. Now, the nicotinic effects apply to stimulation of the skeletal muscles, so we can see twitching of facial, eyelids, and eventually all voluntary muscles throughout the body. Remember that the intercostal muscles are also skeletal muscles, and their paralysis is going to compromise even further the bronchoconstriction at the bronchi. Now, with regards to the CNS, most of the time it leans towards depression, but occasionally you may see hyperactivity and even seizures. So if we ask ourselves, which of all these signs will be lethal to the animal? Well, the answer is the effects on the airways and maybe the heart. The animals tend to die from asphyxia, from bronchoconstriction, and the paralysis of the intercostal muscles. A definitive diagnosis can be made by measuring acetylcholinesterase in the animal, and if it is depressed, ideally you want to identify the insecticide in rumen or stomach contents, or the bait that the animal ingested. If the animal is alive and has signs compatible, whole blood cholinesterase values usually fall below 20% of control values. And you want whole blood because the red cells contribute to the major fraction of the activity in total blood. Now, if the animal is dead, you can test for brain cholinesterase, in brain obviously, and if you do not have means to take out the brain, if it is a large animal, you can send an eye globe. The retina is kind of a specialized brain tissue, and the activity is almost 4 times higher than the brain. Something that can be done on a live animal is giving a test dose of atropine. With a pre-anesthetic dose, you will see an increase in the heart rate, dry mouth, mydriasis. But if the animal is intoxicated, you will not see much of anything unless you give an antidote dosage, which is much higher than the pre-anesthetic dose. For treatment with early or recent exposures, decontamination steps, emetics, activated charcoal with the precaution to avoid aspiration, thoroughly bathing the animal with a detergent if the exposure was topical. What comes closer to an antidote is atropine sulfate, which is not charged and penetrates in the brain. If we use atropine methyl nitrate or glycopyrrolate, these are charged molecules and therefore they don't enter the brain. The dosages are higher than pre-anesthetic dose. Remember that pre-anesthetic doses can be used to make a presumptive diagnosis. In horses, you may want to avoid atropine altogether because of the gut stasis. If we use it, the atropine can be mixed with the fluids and given IV while you auscultate the abdomen. And as soon as there is a reduction in gut sounds, the administration should be stopped. Atropine is usually given at 0.1 to 0.2 milligrams per kilogram, with 1/3 of the dose by IV route and the rest by subcutaneous injection. Ideally, with atropine, you want to revert the lethal signs such as the asphyxiation from both bronchoconstriction and bradycardia. The effects such as meiosis and salivation are not really a concern for survival and should not be used as therapeutic endpoints for atropine administration. There is a drug called pralidoxime that breaks the bond between the OP and the enzyme, and that allows the enzyme to resume its activity. Because the binding is reversible for carbamates, it's only useful for organophosphorus insecticides. And the idea is to resolve the nicotinic signs against which atropine is not very effective. Artificial respiration may be needed to counteract the respiratory paralysis and the hypoxia, which, as I say, is the main cause of mortality. Moving on to nicotine, this is an alkaloid from the tobacco plant and other nicotiana plants like the tree tobacco. Each cigarette contains about 20 milligrams of nicotine, and a pack of cigarettes has been reported to be lethal in dogs. It works by mimicking the action of acetylcholine at sympathetic and parasympathetic ganglia, and also at the neuromuscular junctions of the skeletal muscles and some synapses in the CNS. Now, with high doses, there is a stimulation followed by a blockade, which is what is shown here on the slide. There is a very rapid onset of signs within 15 to 45 minutes of the exposure, and the animal will initially have high blood pressure, high respiratory rate, involuntary contractions, excitation, tachycardia, salivation, lacrimation, vomiting, diarrhea, and this is followed by the opposite: hypotension, respiratory paralysis with very slow and shallow respiration, muscle weakness, CNS depression, bradycardia, collapse, and coma. What is unique about the intoxication is that it's very fast acting and also clear from the body. So if the animal does not die, he recovers very quickly within a few hours of the onset of signs. Because the animal usually dies from respiratory paralysis, from the neuromuscular blockade, the most important therapy is artificial respiration and oxygen. All the nicotiana plants are unpalatable, and animals generally avoid consumption, but there are a lot of cases of poisonings in all animal species. At high doses, the animal will exhibit clinical signs we mentioned on the previous slide, but with low dosages which do not cause clinical signs, the primary problem is teratogenesis. Cattle, sheep, pigs will give birth to neonates with malformations. This slide shows a lamb with a cleft palate on the left and arthrogryposis on the right. And this happens when the consumption takes place during the 30 to 60 days of gestation. Another plant, poison hemlock, is next. This is easily identified because it has purple spots, and hollow stems. The leaves are dissected and look like those of parsley, and the flowers are small, white, in kind of an umbrella-shaped cluster. The leaves smell like parsley when you crush them, and the toxins are piperidine alkaloids, which account for about 1% of all the plant parts. This plant has a history because tea made from the poison hemlock reportedly killed Socrates. Quinine acts similar as nicotine, so it initially stimulates all the autonomic ganglia and the motor end plates, and then depresses them, causing a neuromuscular block. So just like with tobacco plants, at high doses it causes systemic effects, and at lower doses ingested by gestating animals will cause teratogenic effects. This is a paper entitled Toxicosis of Dairy Cattle Exposed to Poison Hemlock in Hay. They isolated the alkaloids in the plants, the hay, and the urine. There were 20 or 30 cows affected from an alfalfa forage contaminated with poison hemlock, and of those 20 animals, 2 died. I wanted to highlight that the ingestion must be massive to result in acute toxicity. In this case, between 50 and 80% of the rumen content was hemlock, and the signs were bloat, hypersalivation, lacrimation, dyspnea, ataxia, and death. The rumen and the urine was positive for quinine, and the treatment they applied was relieving bloat, activated charcoal, and atropine. They gave atropine actually because they thought it was a carbamate or an OP intoxication. Now here is another picture in piglets with congenital skeletal malformation induced by maternal ingestion of poison hemlock. The ingestion might have probably took place between 42 and 61 days of the gestation period. And on this table, the list of plants that are teratogenic in animal species. We have on the left the animal species affected, the period in which the fetuses are more susceptible, and it includes the type of birth defects associated with their ingestion. So as we can see, for poison hemlock, the types of malformation include arthrogryposis. That's when the bones are twisted and not aligned. Cleft palate, scoliosis, torticollis. Another teratogenic plants are lupines. There are about 100 species, and not all of them are toxic. These are perennial shrubs with alternate leaves arranged as the palm of your hand, with about 10 leaflets each one. There are about a dozen quinolisidine and piperidine alkaloids, and they act similarly as nicotine. It is actually one of the most important plants, and as a matter of fact, they cause more deaths than any other single plant in states of the Rocky Mountains. Most losses occur under conditions in which animals consume large amounts of pods in a brief period, and with the small ingestions during gestation in cattle is going to cause crooked calf syndrome, which is what we see on this image. You can notice the forelegs are twisted and they are held in a flexed position. So the last slide is Oncurare. This is a generic name for various South American arrow poisons that are derived from plant species like Cododendron. If we look at the structure, it's a big quaternary amine that induces a neuromuscular blocking effect. So basically what it does, it binds to the nicotinic receptors in the skeletal muscle and competes with acetylcholine, and this is going to result in motor weakness with total muscle flaccid paralysis. The paralysis is flaccid because it's a huge molecule with two charged nitrogen groups. It does not penetrate the brain, and the animal is going to die from paralysis of the intercostal muscles that obviously asphyxiate the victim. So you may wonder what happens to the Indians that eat the prey. Well, absolutely nothing, because the molecule will not be absorbed to any degree in the GI tract. Well, this is the last slide. On the next videos we will talk about more toxicants affecting other body systems. So goodbye for now, and I'll see you on the next one.